let's start it now. Space is so cool, they even have a science cafe. First here we have one cell culturing lab. So here we have one flow cabinet. So in 2021, I obtained my master's degree in toxicology and environmental health at the University of Utrecht. And it was during that study that I found out that I wanted to do a PhD. So then just after my graduation, I just applied for a vacancy for the ZeroPM project here at the Vrije Universiteit. And I was accepted. So yeah, 3 months later, I started here.
We can go to the next lab, which is the heart of the chemistry lab. Where all our working horses are. So I'm an analytical chemist with the years of research in chromatography and mass spectrometry. And throughout my career, I was I have been identifying chemicals in environment, and I was always curious about the risks of the chemicals to the human health and environment. And that's how I ended up in the Timur's lab.
Here we have the incubators, where you can put your cells and expose them, for example, or just for further culturing. So my main objective is to find out the hazards of persistent and mobile substances to human health. I have a whole battery of tests that I perform, and the things that I measure are mainly focusing on endocrine disruption, on developmental toxicity, on immunotoxicity, and on neurotoxicity. In the end, I will determine the concentration at which all these chemicals have their effects on all these cell types, proteins, and embryos.
So here you can see 2 triple quadrupoles that are mainly used for the targeted analysis, for example, of PFAS or other contaminants. My main research objective is to identify persistent mobile substances because we're working in the ZeroPM project. And for this purpose, we use effect-directed analysis. So by means of LCH-RMS suspect and non-target screening, high-resolution fractionations, and a battery of different bioassays, we are able to prioritize and identify these emerging PM substances. My work is integrated in the work package 6 of the ZeroPM project, which deals with the risk assessment of the chemicals. And results of my findings will be integrated of these chemicals, and colleagues from work package 7 will suggest advanced water treatment methods to mitigate pollution of these important PM substances in sources of our drinking water.
So here is the zebrafish lab. For me, the most interesting thing about the ZeroPM project is that we are looking at chemicals that are persistent and mobile. So these are chemicals that have a high probability of ending up in drinking water. And from a toxicology perspective, it's of course very important because, well, we all humans all drink water. And so if the chemicals are in the drinking water, then we are potentially exposed to relatively high levels of those. So that makes it, of course, very important to know the hazards of those chemicals. And to contribute to that, it's, I think, the nicest thing about ZeroPM projects for me.
Okay, and here is another part. It is called fractional mate. The most interesting thing for me to be part of ZeroPM project is an opportunity to contribute to the safer environment and to the reduction of the risk to the public health. And also I find it very nice, and for me too is an opportunity to collaborate with great researchers involved in the ZeroPM project that work in different areas of sciences.
And that machine is for them. I don't know what it does, but yeah.
ZeroPM, Zero Pollution of Persistent and Mobile Substances. This project has received funding from the European Union's Horizon 2020 Research and Innovation Programme under Grant Agreement number 101036756.